Okay, let's start. Okay, good morning, afternoon, evening. Hola, hello, bonjour, hola, ni hao, konnichiwa, 안녕하세요, and hello all, and welcome and thank you for joining us, the 43rd seminar, and I call it Trends in Biotechnology Day. I'm very sorry for rescheduling the 42nd seminar due to the email address change of one of the Rising Star speaker and the resultant difficulty in communicating with him, and also my unexpected flight change leading to my staying at the Israel airport for almost 14 hours just in the airport. And because of that, we need to reschedule. Probably that will be early August. In addition, today, the second schedule change for the first time during the entire 1 year due to my trip schedule. So thank both speakers today for their understanding and for the audience for joining us online with the short notice. Okay, so it is my tremendous honor to introduce very briefly our pioneer speaker, Dr. Matt Pavlovich, the editor of Trend in Biotechnology, a high-impact cell press journal. And Trend in Biotechnology is one of the top review journals, and I often read many papers to learn new areas or the state of the art technology, biotechnology. Matt is one of the most enthusiastic supporters for this seminar series, SynBYSS, and I especially thank him for his dedication to the research community and then his support to synthetic biology. So Matt, the virtual podium is all yours, and thank you again so much for your wonderful contribution to community. Thank you.
Well, thank you. I think it's a too kind introduction. I feel like looking at the list of pioneer speakers, you see Nobel laureates, you see directors of research institutes. I see people whose work inspired me during graduate school, and now to be listed among those people is really a tremendous honor. And I think I'm going to echo a lot of what the other speakers have said so far, in that this series has really been a great way to watch the community develop and really engage with some of the rising stars in it. So I really am excited to be part of it. So yeah, Trends Biotechnology is my journal. A lot of people don't realize that the Trends journals, the review journal published by Cell Press, are a single editor model. And so I do everything in the journal from acquiring content to evaluating proposals, peer review, editorial comments, revision, but also things like journal strategy, going to conferences, meeting the community, keeping up with where the research is in the field. And so it's really a pretty broad job, and I do everything there is to do for the journal. You might be familiar with Cell Press as the publisher of the journal Cell. We also publish a lot of other well-known research journals, Immunity, Neuron, Molecular Cell, and we've recently established a bigger presence in physical and chemical sciences. So we've launched Chem, Joule, Matter, and One Earth in the last 5 or 6 years. So we're really on that journey toward being an all-science publisher, not just a biology publisher. And within Cell Press, we have 16 reviews journals called Trends, and that's been a pretty strong brand for a number of decades. Trends in Biotechnology is unique in that we insist on talking about applications and utility. So I like to call it the leading reviews journal in applied or useful biology. When I say leading, I don't mean just journal impact or other metrics like that, but also really the vision for the Trends journals is as a place for the community to come together and talk about what's important. So both the research and the science itself, but also the practice of science, things like commercialization and translation, ethics, policy, funding, all of those issues that matter so much to scientists, I think we really try to give a platform for. So you might be wondering why it's necessary to write review articles or why this is an important thing. And again, we don't publish any research content. It's just review, perspective, commentary kind of pieces. So, in an age where we can just go to Google Scholar or Web of Science or whatever and search for the term that we need, why are reviews useful? I'm going to hopefully inspire you from a quote. This is from a famous scientist. And the quote goes, "It is certainly impossible for any person who wishes to devote a portion of his time to experiment to read all the books and papers that are published. Their number is immense." You might agree with this. The person who said this quote was Michael Faraday, one of the most famous physicists of all time, and probably one of the smartest people of his age. If Michael Faraday had a problem doing this in 1826, I don't know how any of us are supposed to do it today. And so really, reviews journals, the point is to coordinate and to curate the literature in a way that's accessible for a non-expert. And we really try to minimize jargon. We really try to create something that people in a variety of different research backgrounds can appreciate. We insist on having a novel message, not just this group did this, that group did that, but what's your perspective? What's your expert synthesis? If I invite you to write a review article, it's because you know what you're talking about, and our readers want to hear what you have to say, not just some sort of collection of the literature. There are currently more than 2 million published articles per year in scientific journals. There's at least 30,000 journals. These numbers are a little bit old, so it's probably more than that now. And review and perspective content is a really good first pass at figuring out what's important and what's changing in the literature, and what's important for you to pay attention to. And so I really hope that you understand where we're coming from and why it's important and useful to have a vibrant review community. Synthetic biology is, of course, one thing among many that we cover, but it also manages to touch on a lot of our most important aspects, whether that's metabolic engineering or applied genomics or tissue engineering. There's some sort of SynBio application in all those fields. And so I think this is a really great seminar series to be associated with. I want to conclude by talking about commercialization and translation. And this is something that I've thought a lot more about recently. We publish all these great articles, and I can read about the state of the art of the science and so many fascinating topics. But sometimes what we read about in the journal, you can't go out in the store and buy. So the example that I always give is we know for a fact that you can use algae to make biofuels. That's been established for decades. But you can't go to the gas station and get the algae handle and fill up your car with algae. And why is that? Well, of course, there's challenges related to scale up, process robustness. There's also challenges related to the commercial market, to regulatory factors. And all of these things are equally important to the science if you want what you're doing to make a difference. A couple of statistics I found. In 2016, this is the most recent year I could find the statistic for, about 2% of all patent applications, or patent grants, I should say, were to academic labs. So it's about 6,000 out of 300,000. And among those, only 5 to 10% actually go on to be commercialized. So that's a few hundred inventions per year coming out of academic labs that actually make their way into the commercial market. And I'm not saying that your work has to have commercial relevance to be important. Obviously, things like fundamental science and mechanistic work, those are critical to underlie the technologies that we develop. But at the same time, I think it's increasingly important to think about, if I'm going to make a technology in the lab, how do we get it out there? And if we say this has this impact to reduce climate change, or to help food security, or to improve human health, how do we actually make that happen? And I think that's what I want to end on, is really impressing upon people who are doing work in these fields to say, not just this is my motivation statement to get an NSF grant or to get published in a high impact journal, but really this is the driving force behind what I'm doing. And this is why I want to get it out there. And then this is my path to do it. And so I think if you can think about that as you're doing your work, I think your science is going to be even more impactful.
So amazing. 100% agree with you. all the problem we currently having, including food inequality or climate crisis. I'm seeing temperature right now 40 Celsius, but Madrid I'm currently at, we had 47 Celsius. And also you're talking about medicine, those absolutely important, and translation should be something we need to deliver the real product after more than 20 years of synthetic biology, the field study. So it's time to deliver, and then absolutely we need to do something for the most practical one. In that way, public understand scientists better. And also, absolutely, you are right. I need to confess, I do not read the individual paper anymore because I only read abstract, title, that's it. And still too many, paper to scan. And the only time I study or do research is when I need to write the review paper, or when I need to write the proposal. That's the only time I read the paper, individual paper. And to do that, I go to review journal, like your journal, and then I just skim to learn new thing. And then I go to individual papers, and then absolutely review paper, reading the field. In that way, people learn new thing. And so your job is absolutely important for the community. So thank you so much for your wonderful talk. And I absolutely agree with you. Thank you. Okay, so now the main speaker of today with a longer introduction. Gözde Demirer was born and raised in Istanbul, Turkey, and received her bachelor degree in chemical and biological engineering from Koç University in 2015. In fact, I was in that city 1 week ago to enjoy my weekend with amazing food and cultures of both Asia and Europe. The diversity was absolutely wonderful there. She completed her chemical engineering PhD at UC Berkeley with Professor Laundrie in 2020. And during her PhD studies, she developed nano and biotechnologies for plant genetic engineering. As you can imagine, now plant engineering, genetic engineering emerging and then become hot topic, but very important, but not many people working on that, but she is one of them. So I call her true rising star. For her postdoctoral work, she joined Professor Brady's lab at UC Davis, where she studied nutrient use efficiency of tomato and developed functional genomics and synthetic biology tools to study transcriptional regulation. And as you can imagine, plants are too difficult or too challenging to engineer, but she is brave enough to enter that one. In my career, I may not enter plants synthetic biology. She has just started her faculty position at Caltech officially 1 month later as an assistant professor, and she will keep working on bioengineering of plants for food security, very important, sustainability, another very important one, and environmental remediation, and all absolutely great topic. She's currently recruiting, so feel free to reach out to her after her talk today. And I truly believe she is one of the true rising stars, as I told you earlier, with a huge potential. So if you think about your career in a great lab, join her lab. And I will tell you, one of the greatest scientists, Professor Francis Arnold, love her, period. Okay, please take it away. Thank you.
Thank you so much. Let me get my slides up. Okay, does it look good?
Yeah.
Okay. Yeah, thank you, Taisok, so much. That's a very kind introduction, and thanks for having me here. I think it's really amazing what you're doing with the seminar series. And I can tell you that it's definitely been the highlight of my week over the last year or so to hear the new research from the new young folks, new faculty, and also from getting perspectives from the leaders of the field. So thanks again for this amazing initiative and including me in here. Also want to say hi to everyone who's joining us online today. Thank you. As Taisok introduced, my name is Gözde Demirer, and I'm currently a postdoc in the Brady Lab at UC Davis, and I'm about to start my own lab at Caltech Chemical Engineering this September. So my lab's overarching aim is to improve both the plant and the environment health using interdisciplinary approaches from the fields of chemistry, material science and engineering, biology and bioengineering, and also many more fields. I also want to quickly call everyone's attention to the newly restructured Resnick Sustainability Institute at Caltech, which will be tackling many difficult problems of climate change and sustainability. We have a lot of resources, new facilities, cutting edge equipment, together from faculty coming from all areas to collaboratively address the important sustainability challenges. So Caltech is really an awesome place to be right now if you're interested in making positive impact in these areas. And as Taisok also mentioned, I'm currently recruiting, so please do feel free to reach out if you're interested in these areas. So today I'm gonna start with what has been motivating my research so far, and that is to enable future food security using sustainable approaches. Given the global population growth and our increasing food consumption, it's predicted that we need to increase our food production by at least 60% by 2050 to meet this growing food demand. However, it's also estimated that our water supplies are going to be 40% less at that point. Now, unfortunately, the water is not the only compounding effect. We also know that the climate change is and will have extended and expanding negative effects on our agricultural production values. There's a recent NASA study that was done in 2021 that estimated a major decline in our stable crops, as shown here. And this is by 2030, and it's based on the temperature and water availability conditions that we expected to have. So these are some abiotic stresses, but on top of these abiotic stresses, we are also dealing with some new emerging plant pests and diseases due to the increased temperatures early in the crop growing season, as well as the shifts in the season lengths. So currently around 40% of our crops per year globally is lost to these diseases, which is also expected to worsen with the new pests emerging given the climate change. So far, we have been using a lot of unsustainable approaches to meet this growing food demand. And majority of these agricultural practices that boost the plant yields actually ending up harming the soil, environment, and the organisms that live in them. Excess chemical fertilizer and pesticide usage is one of these practices. And in fact, we use more than 400 billion pounds of fertilizers and 1.5 billion pounds of pesticides every year. And it's said that only around 1% of these chemicals do interact with their target organism. So nitrogen and phosphorus are the most common fertilizers that we use, as these are vital nutrients for the proper plant growth, as shown here. Nitrogen fertilizers have been obtained through this Haber-Bosch process, which is an energy-intensive process, while the phosphate fertilizers has been mined through the natural phosphate rock reserves. However, there are drastic consequences of this excess usage of chemical fertilizers. In the case of nitrogen, the Haber process consumes around 5% of world's natural gas production and around 2% of our energy supply, which is really high if you think in the broad sense. And then the runoff of the nitrogen fertilizer causes eutrophication, atmospheric deposition of nitrates and ammonia, and also higher emissions from agricultural fields of nitrous oxide is now also the third most important greenhouse gas following the carbon dioxide and methane. And in the case of phosphorus, it's estimated that the rock reserves are going to deplete within 20 to 50 years. And then the excess usage also has similar negative effects of eutrophication due to toxic algae blooms and soil deterioration. An alternative approach to increase crop yields and to provide resiliency to climate change is through plant genetic engineering. which is a way more sustainable practice and has been showing great promise so far in agriculture. However, there are several bottlenecks of plant genetic engineering that really limit or hinder its widespread usage and its ability to reach its full potential. So the first bottleneck is actually the gaps of our knowledge in plant biology and plant genomics. Just to say the least, plant genomes are very large, and what I recently learned is that their gene expression regulation is very complex, to say the least. So we had a lot of advances in next generation sequencing. So we have full genomes of many plant species nowadays, but the functions of most genes are still unknown in plants. And this is partly due to the high functional genetic redundancy we see in plants, meaning a lot of proteins doing the same job, but also I think partly because we are lacking effective tools to make mutant plants to study the function of these proteins. So plant biomolecule delivery and transformation tools that we have currently, they are highly species dependent, very label intensive and time consuming. So it limits a lot of labs' ability to carry out these studies. Last but not least, there are still many hurdles to transition these engineered plants successfully from lab to field and to markets, as Matt was mentioning. And I think some of these are due to the regulations of genetically engineered plants that change over the globe. But I would also claim that more importantly, we still don't have a complete understanding of plant genes and genomes under the actual field conditions when plants are in close interaction with the environment and other organisms surrounding them. And then finally, there are still, unfortunately, many societal and political barriers for the widespread usage of improved plants. So today I'm going to focus on this first bottleneck and talk about my postdoc work that is titled Conservation and Repurposing of Nitrogen Regulatory Circuit between Arbidopsis and Tomato. And I will also describe the tools that we developed to study plant transcriptional regulation. These tools that I'm going to talk today, they are in no way specific to nitrogen circuit nor plants, and they can be applied in other organisms as well. A quick note here for non-plant folks, Arbidopsis thaliana, is the model lab plant that is used in many genetics and genomic studies so far. And we routinely try to translate findings or gene targets for improved traits from this model plant to other crops. Therefore, it's really important to study the conservation between this model plant and other crops, such as tomato in our case, for future translation of these technologies to field. So I would like to first introduce the team and acknowledge the people who did this work. This has been a very large collaborative project between the labs of Nicola Patron at the Earlham Institute in the UK and the labs of Siobhan Brady and Dave Siegel at UC Davis. Arbidopsis work has been carried out in the Patron lab, and then the tomato work has been carried out in our lab together with Chabian, another postdoc in our lab. And today I will only have time to cover a small portion of this project, but there are also very exciting new parts that we are finishing up, such as the epigenetic manipulation of the nitrogen circuit. So if you're interested in this topic, please keep an eye out for a paper towards the end of this year on this project. So our overarching goal in this project is to generate tomato plants with optimized nitrogen use efficiency, meaning we want to have higher yielding plants with less fertilizer input, as motivated in my earlier slides. However, the first issue we face is that there are no molecular players in tomato for nitrogen signaling, or its transcriptional regulation. So we don't really know what happens at the molecular level from when there is nitrogen deficiency in soil until we start seeing these starvation phenotypes. Additionally, most plant traits, such as nitrogen uptake and use efficiency, these traits are controlled by very complex networks, multiple genetic factors interacting combinatorially in plants, and we typically see compensatory effects or genetic redundancy when we look at these complex traits in plants, which makes it really hard to find the correct genes to manipulate when we want improvements. So if we want to develop high yielding crops with less nitrogen fertilizer input, we first need to be able to understand and then predictably manipulate these networks of nitrogen regulatory genes. And then specifically, our goals here are to first generate a predictive model of this nitrogen regulation in Arbidopsis and tomato, and then to functionally validate this model and then compare these networks in between these two species, and lastly, to perform rationally designed CRISPR-Cas9 mutations for increased nitrogen use efficiency in tomato. Doing this way, we believe we can achieve a precise manipulation and fine-tuning of gene expression, and we can also get more complex expression patterns and improved capabilities that wouldn't be possible by just manipulating the downstream nitrogen genes instead. So we think that these are the advantages for manipulating the gene regulation rather than manipulating the genes themselves. So today, the outline of my seminar is that I'm going to first walk you through the process summary that we followed to get these predictive network maps. And then I'll discuss in more details the tools we used and then the tools we developed for this project. And then lastly, I'll show you an example of how we ended up using and combining all of these tools to dissect interactions and to come up with the predictive network model. So the project started with the identification of putative transcription factor binding sites using yeast-1 hybrid and DEB-seq assays in Arbidopsis. These two parts were carried out before I joined the project, so I will not go into the details of these methods, but I'm happy to chat after the talk if anyone is interested. So after we have the predictive binding information from the yeast-1 hybrid and DEB-seq, we initially used an in vitro well plate-based method called TRAMP that was developed by the Patron lab. And then this in vitro validation was followed by the in vivo binding validation. And we have done this in plant root chloroplasts using a method that's adapted from this earlier paper that uses glucocorticoid receptor tech transcription factors. And then, as I said, this is done in protoplasts, and protoplasts are plant cells that are removed from plant tissues without their cell wall. So these are cells in solution, just like animal cells. So they enable high throughput applications in plants. Using this method that we call TARSIT, we can not only validate the binding in planta, but we can also determine its regulatory consequences. So meaning whether the binding actually activates or represses the target genes or has no regulatory effect. So I'll be talking about these TRAMP and TARSIT methods that we developed. But in the summary, using these two methods, we then created a network of transcription factors that is predicted to play a key role in nitrogen responsiveness, together with their predicted regulatory interactions. And then we use this network map to create knockout mutants of these transcription factors in the network in a single and higher order manner to be able to understand their role in nitrogen response. We first generated tomato hairy root mutants, which I'm also going to talk about, as well as stable mutants in Arbidopsis and tomato. And then lastly, we are in the process of phenotyping these mutants, both at the molecular and anatomical level. I developed a cool tool that I call Parrot. That's a dual luciferase reporter assay that's also root protoplast based, and that can detect the overall response of these transcription factor mutants to nitrate availability in the soil. And then as a note here, tomato plants can take up both ammonium or nitrate as a nitrogen source. And under the field conditions in California, where we grow most of the tomatoes in the United States, ammonium is actually quickly converted to nitrate in the soil. So we only considered nitrate as a nitrogen source in this project. And then we are currently completing this project by looking at the anatomy of select tomato mutants for root system architecture, nitrogen use efficiency, and other fruit-related agronomic traits. As I mentioned, this project started with a yeast-1 hybrid assay in Arbidopsis, which predicted this nitrogen network you see. And I know it's too small to read or see anything, but the pink things in the center here, these are transcription factors that regulate many downstream genes as shown at the periphery of this network. And then you could see it's a very complex network with combinatorial interactions. And in fact, there are more than 1,600 interactions between 431 genes, 345 transcription factors, and about 100 promoters. So now you can have an understanding of when I meant complex, what I was talking about. And then within this network, previous Brady Lab people were able to identify a critical 7-member feed-forward loop that was predicted to have a key role in nitrogen response in Arbidopsis. And then with Patron Lab, they used DEB-seq to refine these interaction and binding. And then they identified putative transcription factor binding sites. So you can see here, there are many transcription factor binding sites on each transcription factor in this network. And then also there are some nitrogen metabolism genes that they considered, like this nutrient reductase 1, NUR1 here. So we want to transition this network to tomato, which we didn't have tools to be able to do the, DAP-seq and other methods in tomato. So we take this network from Arbidopsis and then we translate it to tomato by first finding the homologs of these transcription factors using the phylogeny maps that we created in our lab. And then you could see that it seems like this A-T transcription factor does not have a homolog in tomato, which already signals us that this nitrogen network may be wired differently in tomato. And after finding these homologous genes, we used an online tool called FIMO that could do motif detection to predict the transcription factor binding sites in the tomato promoters for those transcription factors of interest we have in our hand. And as you can see, FIMO predicted quite a few binding sites for each transcription factor. And then Patron Lab developed this in vitro assay that we call TRAMP. So TRAMP can validate these binding sites that we found through FIMO and through Arbidopsis DAP-seq. So how this assay works briefly is that we have a DNA probe with a putative binding site in it, and this probe is attached to the well plate surface. And then we can have transcription factors tagged with HYBID tag. We can express these transcription factors in E. coli and PRUFI, and then we can incubate the transcription factors with the DNA probe in the well plate. And then after incubation, we wash away. So after washing, if there is no binding, the transcription factor will be removed from the well plate. And then if there is direct binding between these two, the transcription factor will still be in the well plate. And then we can add the HYBID reporter, which complements the tag and then gives us a luminescence signal. So if there is DNA bound to the transcription factor, we'll get luminescence signal. In the other case, we do not. So we validated and benchmarked this assay with a more common tool of electrophoretic mobility shift assay, EMSA, to make sure that the assay works correctly. And it's a really nice assay because it requires very minimal protein to be able to do it. And it can quantify different probe. You just need to change the small sequence here. And then you don't need next generation sequencing or any sequencing unlike the DAP-seq method. So I will not show the actual data here as it will take a long time for us to go through all these interactions, but I'll show some representative results later. So using this TRAMP tool, we were able to validate some of these predicted sites as shown here on this side of the slide. So the fact that we have binding does not mean that we will have gene regulation because binding does not always result in gene expression changes. So we also need a method that can validate these binding in planta in vivo, but also that can tell us if it has a regulatory consequence. So for this, we developed a method called TARSIT that was adapted from Bergman et al. Previous paper to identify the regulatory consequences of transcription factor target interactions. So how this works is that we transfect the root protoplast with a glucocorticoid receptor tagged transcription factor, which allows the control entry of this chimeric protein into the nucleus by the addition of GR ligand dexamethasone, DEX. And then the effect of the gene induction by DEX treatment can be studied in the presence or absence of cyclohexamide, which is a translation inhibitor that allows us for the distinction of direct and indirect targets of this transcription factor of interest. So the steps are basically transfection with the GRTF constructs as well as a calibrator rupture to normalize. And then we do treatments with nitrogen and DEX and or cyclohexamide. And then we can extract RNA and perform qPCR on the suspected direct and indirect targets of this gene. This is a really sensitive method that gives us implanta information, which is really important to check these implanta. And then, as I mentioned, it doesn't only distinguish between the direct and indirect, but it also tells us what's the direction of this regulation, meaning if it's an activator or a repressor of the target gene. The only downside is that it does rely on an efficient transfection. And if that's not the case, we would need to sort these cells with FACS. So there's a way to overcome that limitation as well. So now that using these tools, we can generate a network, the next step is to actually generate the knockout lines of these transcription factors in the network so we can assess their overall role in tomato's nitrogen response. So we use this loop assembly and cloning approach to generate the CRISPR construct to make the single and higher order mutant combinations in this network. In tomato, we typically use 2 to 3 guide RNAs per gene, per transcription factor. And we had constructs targeting 9 genes in some cases. So that's 27 guide RNA cassettes in one vector. And I could say that the loop cloning is really great for these type of cloning applications because we had no problems with this. So after cloning, we first decided to make hairy root mutants to dissect the network rather than doing the regular stable transgenics. This is to save time and cost. So how the hairy root transformation system works in a way that we start with a bacterium called Rhizobium rhizogenes. This bacterium is capable of transforming the cotyledon pieces, these are embryonic leaves, and then also generating mutant roots from these leaves, which is like a fascinating technology. And then you can culture and propagate these roots on plates and then with an antibiotic resistance, and you can genotype them, and then you could keep propagating your homozygous lines and for subsequent studies. So I want to compare this with stable transformation for plants who are not, for people who are not familiar with plant transformation, is that this is really a quicker, easier, and more affordable way than to do stable lines. So in tomato, how we do it typically takes around 12 months to get the transformed T0 plants and 18 months until we can have homozygous plants. You know, my postdoc was 2 years. There's no way I could do this project if I were to do stable lines. But in hairy roots, we can get everything from start to finish for homozygous lines in 2 months. The problem with this is that the hairy roots technology is only developed for certain plant species. Good that it exists in tomato, but it doesn't exist in everything. And then you could imagine that you can only study root-related or cellular-related traits and not all the traits using the system. And then lastly, after we have the mutants, we want to be able to study their effect on the nitrogen response of plants. So I developed this tool that is based on dual luciferase reporter that tells us the effects of these transcription factors. So how it works is that we grow the tomato mutant hairy roots and Arbidopsis mutant roots on plates, and then we extract root protoplast, and then we transfect these root protoplasts with a set of luciferase vectors. So here, one vector expresses ELUC, this is a green luciferase, under the control of an NRP promoter. So this is a synthetic promoter that was discovered in Arbidopsis, and it is responsive to nitrate. And then we have another vector that expresses a red luciferase under the control of a constitutive promoter. And this serves as our calibrator between different batches of protoplast and different batches of transfection. So this assay is really scalable. Depending on your needs, you can do test tubes, you can do well plate. After the transfection, we incubate it for 1 day under different nitrate conditions. And then we can check the ratio of green to red luciferase using a plate reader. So it's a really high throughput, rapid and quantitative method, and it's really scalable to test all of different nitrogen conditions, mutants, different time points. So it's been really helpful for us to phenotype these mutants. So most of the protoplast assays in plants are actually done using leaf protoplast because it's easier to get leaf protoplast. And then it's also, even when you can get the root protoplast, it's been hard to transfect them with high efficiency. So early on in this project, I spent some time to optimize the protoplast protocol so that we can get protoplast from the tomato roots, and then we can transfect them with efficiently. So I'm showing from just 3 regular Petri dishes, we can get more than 10 to the 6 cells with high viability, and we can transfect them with high efficiency. So here I'm showing the expression of cytosolic GFP as well as nuclear GFP in these root protoplasts from tomato. And then I want to come back to the synthetic promoter that I mentioned, NRP. So this promoter was discovered in Arbidopsis in 2010, but we also wanted to make sure that it will work in tomato. So what we did is we created these hairy root lines that we used NRP promoter to express a nucleolide localized GFP. And then we could see that the GFP expression gets higher at higher nitrate amount. So it seems that this promoter also works in nitrate as a proxy to nitrate responsiveness. We also wanted to test that it works in protoplast. So we extracted protoplast from the wild type Arbidopsis and wild type tomato plants. And then we treated them with different nitrate conditions after transfection with the dual luciferase system. And then we see a similar trend where there's more nitrate, we see more expression. So this verifies that we can indeed use the NRP promoter activity as a proxy to nitrate responsiveness in tomato. Okay, so now I'm finally going to demonstrate how we ended up combining all of these developed tools to be able to dissect the nitrogen regulatory network. So this was the feed-forward loop that we are hoping to dissect and functionally validate. And I'm going to show an example of these NLP7 transcription factors and their interaction with the NIR1 downstream gene and promoter. So NIR1s and NIR2s, these are nitrate reductases. They are involved in the second step of nitrate assimilation. And you can imagine that their expression is induced when there's nitrate. So we first use the TRAMP assay. This is the in vitro assay that I mentioned. This can assess if the NLP7 transcription factor binds to the promoter of NIR1. So we already have some putative sites predicted from the DAP-seq, and we tested several DNA probes, and then also a negative control, which is a random DNA sequence that does not have any transcription factor binding sites on it. So in this case, we could see that this probe 1, the probe that is closer to the transcription start site, had a positive binding interaction with the NIR1 promoter. After verifying this binding with TRAMP, then we move on to our in vivo assay, TARSIT, and then see if it binds in vivo and if it has a regulatory consequence. So here we are using NLP7 transcription factor fused to the GR, and then after induction and nitrogen treatment, we are looking at the gene expression levels of NIR1 using qPCR. And we do see that there is a direct activation of NIR1 by NLP7 that also confirms the binding from the in vitro assay, and then also tells us that it's an activator in this case. And then lastly, we move on to our last assay, PARROT, and then work with the mutants of NLP7 transcription factors. And then from TARSIT data, where NLP7 seemed to be an activator, we would expect to see a decrease in the mutants of the nitrogen responsiveness. And that's what we see with the PARROT assay in both the single mutants and the double mutants of this NLP7. So if we combine all these results, then for this network edge, we can say that NLPs are direct binders of these nitrate assimilation genes, and then they activate the nitrogen response. And this has been shown by us to be true both in Arbidopsis and tomato. So this is just an example for the NLP7 edge. So then we go back and we do the same analysis using all the tools for all the edges that you see in this network. So given the time limitation, I won't show all these edges, but I'll show you what at the end our predictive network looks like. And then this is how it looks like. So this is the nitrogen regulatory network in Arbidopsis that we put together using all these assays that I showed you today. And hopefully you can see that it's a very complicatedly wired network. So I'm gonna reduce this. I'm gonna show you a simplified version, both in Arbidopsis and tomato, where I removed some of the edges and interactions so that we can look at it closely. And I want to show you two examples, what this data means to us. So first, let's look at DREP26 here. So our results show that the NLPs are positive regulators of NIR1 and NIR2. And then our results also showed, which I didn't share today, but DREB is a negative regulator of NLPs. So then one positive and one negative interaction, you would expect DREB to be a repressor of the nitrogen response to have a negative regulatory effect. And then when we look at our PARROT assay, when we look at the mutants of the DREB, you could see that they have increased response, validating DREB transcription factors, negative regulatory role in nitrogen response in tomato. So now this is a great target for CRISPR editing, for example, to increase the nitrogen responsiveness. And then the other example I want to look at is ARFs. So following these regulatory interactions that I'm showing here, we would expect the ARFs in tomato to be activators of the nitrogen response, as we have 2 negative and 1 positive regulation. It's like a little puzzle. And then in Arbidopsis, in opposite, we would expect them to be repressors because we have 2 positive and 1 negative regulation. And then this is exactly what we see in the PARROT assays, where tomato mutants have reduced responsiveness, while Arbidopsis mutants have increased responsiveness. So I have several comments here. First is that the role of these NLP transcription factors in Arbidopsis was known in literature before. So the fact that we are getting matching results with literature validate all these new assays that we develop. And on top of that, we were able to show that this role is conserved in tomato. On the other hand, the role of ARFs in nitrogen regulation was not known. ARF stands for auxin response factor. Auxin is a very important plant hormone. And it seems like these transcription factors also have a critical role in nitrogen response that we didn't know before. And then our assays did not only discover this interaction for the first time between the auxin and the ARFs and the nitrogen pathway, but also showed that the regulatory function of this transcription factor is diverged between Arbidopsis and tomato, which is very important for downstream crop improvement applications. So what we are doing right now, after having these networks established, we are using the same vectors that we use for hero roots, and we are making stable transformations of some select transcription factors and their combinations in Arbidopsis and tomato. We have genotyped these lines, and we are in the process of phenotyping these lines. And so far, I'm excited to tell that we are seeing the predicted phenotype in these rationally designed mutants. So with that, I'm going to conclude here and say that in collaboration with the Patron Lab, we developed several tools to generate a predictive nitrogen regulatory network in Arbidopsis and tomato. Our results show that there is both conserved and diverse parts between these two species. And then we believe that the predictive network model that we established is going to help with rational genetic engineering of tomato for increased nutrient use efficiency. So I'd like to thank everyone in the Brady Lab, my postdoc advisor, Siobhan Brady, and also Chabian, who I worked very closely on this project, and the team of undergrads who helped us. I would like to thank, of course, Nicola Patron for their help with the essays and the Arbidopsis part of this work, and our funding sources, USDA, NSF, and BARD, and Resnick Institute at Caltech. So if Taisok lets me, I'm going to take one more minute, come back to the bottlenecks slide, and then also give a quick note to everyone that during my PhD at UC Berkeley in Landry Lab, as Taisok introduced, I worked on developing technologies, nanotechnologies to develop cargoes to plants. This is to do transient expression in plants that can have permanent CRISPR gene edits. And we think that this tool is really cool and very useful because it can be used in a plant species independent manner, and it can greatly facilitate plant transformations with minimal regulatory burden, as it does not rely on any bacteria or transgene insertion. So we are super excited about this technology, and I do hope to keep on working both on the nanotechnology things and the synthetic biology and bioengineering things. We also have very exciting new projects in environment and rhizosphere engineering, plant microbe interactions. So if any of this sounds interesting to you, here's my email address. Feel free to reach out to me. And I'm also happy to help folks who want to get into academia or get a, have their own lab. So thank you all for listening. I'm looking forward to the Q&A session.
Amazing, amazing. So, I mean, amazing like the last time when I listened to your talk. So again, I mean, you are so brave to do plant engineering despite all the challenges and waiting time, I mean, for data and for many months. So thanks for developing those fantastic tools for the community. And again, I mean, I now clearly see why Professor Francis Arnold loved you. It's all great, the science and engineering. I mean, that's amazing. Also, I realized I study every week by listening to such wonderful talks, minimum 1 hour a week for 1 year, rather than I said, I'm not studying at all, and every speaker covers many papers and important topics in 1 hour talk. So I do study and spend time to understand the hot field, and every week by extensive but concise summary. So thank you so much. And let me see whether we have questions, but in the meantime, Matt, do you have any comment, challenging question while I'm waiting for others?
Yeah, so you mentioned briefly that part of the motivation for this work is to look at targets that you might be able to affect using CRISPR or some other gene editing technology. What do you think is the most promising target that you've discovered so far? And what are maybe some challenges in actually manipulating it for real life plants?
Mm-hmm. Yeah, great question. So I guess one thing we're still in the process of is to look at the fruit traits, which we haven't done. So I think it would be hard to give you a target without knowing how it affects the fruit, right? Because in the case of tomato, that's the most important thing. That's the part that humans that we eat. So we need to make sure that whatever the mutant that we go forward with is not reducing the fruit quality or taste and all the other things that I didn't know people care about, if you could process it for ketchup and things like that. So far we've been only looking at root traits and these like reporter assays. And from that, the reason why I showed DREB is I think it's one of the great ones because it's increasing the nitrogen responsiveness. So you would imagine that plants are going to be able to either sense quickly that there's nitrate starvation or act quickly to activate the downstream genes. And then it's also, there are other studies showing that it, in some cases, help with other abiotic stresses as well. I realize more and more that these things are always connected, that when you're improving for sometimes nutrients, you're also improving for water scarcity or high temperatures and things like that. So we think it could have other benefits as well. But then we need to go back and look at the fruit phenotypes. And I guess the challenges I mentioned a little bit, we kind of cut a short variant of the hairy root. Now we are doing the stable mutants, but in tomato, there's a way to do it. It just takes long time and a lot of work. But then if we are trying to extend these type of work to other crops, we are very limited in which plants we can do this and which cultivars we can do this. So there are like a lot of different cultivars of crop species. And then sometimes you get one working, but not the elite one that the people like. So I think most of the challenges from going from lab to field, some of them comes from the transformation challenges. And then the fact that if you use the bacteria to do it, you need to segregate out the transgene because otherwise it's a GMO. So I think that kind of motivates the tools that I developed in my PhD as well. If we could do this without getting the transgene in there in the first place, that helps with the regulation, and if we could reach out to plant species that are not reachable by the common transformation methods, I think that's other important areas that we can work on.
Yeah, that's a great answer. That was actually anticipating my other question too, which is how broadly generalizable is this? I know that, for instance, some people are interested in fast-growing grasses as a biomass source for fuels or other chemical production. So if there's a way to easily identify targets for improvement in those kinds of plants, I'm sure that can go a long way too.
Yeah, I mean, the regulatory network tools that I talk, as I mentioned at the beginning, all you need is a cell and a way to transfect it. So not only grasses, but you could think of doing it in any cell type of interest. So I think the tools in that regards are very translatable, less so with the transformation technologies, right? So that's going to be challenging for certain grass species. Like I mentioned, in the Brady lab, we also work with sorghum, and then that's a huge another challenge to be able to transform sorghum in any way. And then even in tomato, right? So all the results that I showed today is with domesticated tomato called M82. It's actually the variety we use to make ketchup. So we get funding from Heinz, for example. But there are varieties of tomato that are more resistant to like salt, for example, and drought, that are like wild varieties that are grown in South America. We cannot transform them. We cannot even get them to flower in our growth chamber. So even within species, we have a lot of limitations of translating this to other plants. But I also want to echo the point you said, like that we always need to kind of keep in mind, how do we take these things to market to people if it has applications. As you said, it doesn't need to, but I feel like in our case, as academics in this field, there's a lot we can do about the technology development, but there's also a lot we can do to engage the public and society, because with GMO foods, it's there, even though now these are not even called GMOs anymore, they're called genetically engineered GE foods, but you still get a lot of resistance from public and regulatory agencies. So I think we shouldn't just stop there. Okay, I did the science, this is the technology, and then it's not going to go anywhere unless you actually push your way through those other more public and societal and economical issues of like seed distribution to other companies other countries where it's most needed. So I think there's a lot that I'm also excited to be more involved in as I go through my career.
Yeah, I absolutely agree. I think one thing that I've really come to appreciate since I've started at the journal is that the regulatory and the public perception situation is just vastly different in different countries. Mm-hmm. Even within Europe, where there's European frameworks, that sort of thing, how it's implemented in different countries is different. And then how the consumers react to it is significantly different.
Mm-hmm.
Yeah,
absolutely.
That's absolutely. So actually, that was my question I wanted to ask. So again, the brilliant answer, and then also coincidence somehow, because I gave a talk today in one of the Madrid center, and I'm talking about lycopene, and lycopene coming from tomato, as you can see. So I'm making my lycopene from the plastic waste. And then we are talking about similar thing. And also we're talking about GMO, but I do not use the term GMO because GMO has hugely notorious bad meaning to the public. And I stopped using that word because now I use the genetically engineered microbe. I mean, you mentioned genetic GE organism. I call it GEM, genetically engineered microbe, and that is GEM, the one of the best thing my wife wanted to have. But after that, I have less problem in terms of one. But I wanted to ask one challenging question, before we end. So, if someone like Bill Gates give me, let's say, billions of dollars to solve the climate crisis by doing anything without any restriction, limitation, my answer to him or to her, I would ask people to stop the development of cities and factories and plant trees all of the cities. But this strategy is not practical at all. So again, plants are the way to go by engineering them. But at the end of the day, I mean, we face GM or GMO issues again, especially in European countries. So could you kind of comment a little bit more on this challenging point? That's not even a scientific question, but more challenging question, I believe.
Me or Matt?
No, you.
Okay.
This is your day, right?
Okay. Yeah, I mean, it's a great point. And you said climate change, so it's a very big area, right? So I think there doesn't need to be only one perfect solution. We should take action on many things. And I do believe, as you said, plants should be the key effort in that. But I would say, and I just re-emphasize what I've been saying, I think we can do a lot more if we had tools to be able to transform them easily. There are so many crops that, or not only the crops, but the wild plants, all the plants in all parts of the world, like you can think of that if we could have tools to manipulate them, that there's a lot more we can do. So I think one of the biggest, not only the biggest challenges to be able to work with all the plant species. So if I was given like limitless amount of money, that would be the first challenge that I would focus, because once you can work with these plants that are drought resistant, that grow in rocks, like no water, no nutrients, and they are fine, and you could make them be more nutritious, you can grow them in US or in other parts of the world, then you could solve a lot of problems. It's of course tangentially related to sustainability, climate change, but also food security, right? Because we have a lot of temperature switch, water availability switch, and then as I said, the wheat you grow here, you're not going to be able to grow within like 20 years because of all those conditions. So we need to expand to other crops, but we can't. So I think that's the biggest challenge I would address.
Yeah, that's wonderful. So the reason I try to produce lycopene using the plastic waste is, to me, I mean, my dad also said the food, I mean, never play with food. And then, but many people in America, especially, they too busy or lazy, so not eating tomato just as a food, but they try to get the pill supplement that containing lycopene, and then that many Chinese company actually extracting the lycopene from the tomato. So that's absolutely not acceptable, because at the same time many people in Africa die because they don't have food. So I'm going to try to, solve the problem, in terms of pollution, and also at the same time, the food inequality. And then I believe you have the better, tools to make the more food, from using, the tool, you develop. And then I absolutely thrilled to see more out of your technology. So thank you so much. That's absolutely wonderful.
I think you gave a really great answer to that question. I think if I could maybe generalize that a little bit, I think my answer to that question would be to improve CO2 fixation and sequestration among a variety of organisms, whether that's plants or fungi or microbes or whatever, and use that to produce things of interest or of use for human society, whether that's nutrients or food or fuels or specialty chemicals. Right. I think there's a lot that we can do. And if we had those billions of dollars you're talking about, I think you could, as you're saying, transform a lot of different kinds of organisms. Right. Figure out what works best in what organism, figure out, your efficiencies and your inputs and everything else, and then use that as your primary means of production.
Right, exactly. So, one example, Tech, recently, demonstrate the beautiful paper, about the CO2 conversion into some other, thing. That's absolutely, brilliant one. The only worry I have is, that's still, so-called GMO. And then I do not worry about GMO much in terms of biocontainment. Because most of the GMO or GM, not competitive in terms of, when we deploy into the environment, except one organism which have higher efficiency of CO2 fixation and photosynthesis, because that's giving the fitness benefit for the. So we need to consider that part, how we're gonna implement the biocontainment, but we need to have those organisms that has a higher efficiency of CO2 fixation to solve the problem. Absolutely right. So I guess, we now already passed the time, 6 minutes. So I want to conclude. We can still kind of continue to discuss, after this one. So thank all for joining and staying today, especially during the vacation weeks. I heard many European and US colleagues continue to enjoy their vacations. And we will meet again next week on July 28th, Thursday, the same time, the same Zoom link. And now it's no more scheduled on a Thursday. After 10-day break, I'll be joining from my home finally after my crazy 10 weeks of seminar trip. Although I will resume my trip again on that day, and mostly domestic trips, including Texas, San Diego, San Francisco, Berkeley, etc. We'll have Professor Paul Freeman from Imperial College London, where I gave my talk a couple of weeks ago, and Dr. Sudeshna Mana from University of Utah. And as usual, the follow-up informal chat will occur without recording. So please stay here if you are interested in chatting with today's speakers. And I will stop recording. Give me 1 second to stop record. Stop recording.